The Drosophila protein Sentin binds to EB1 at the plus ends of microtubules, enhancing their dynamics by increasing both microtubule growth rates and catastrophe frequency. As Hiroyuki Ohkura from the University of Edinburgh explains, however, depleting Sentin has little effect on the mitotic spindle, at least in Drosophila cells cultured in vitro.
Therefore, we want to know the real function of Sentin. So we made a Drosophila mutants. And this mutant is perfectly viable with a minimum impact on mitosis and male meiosis. But it was female sterile. So we looked at the Drosophila oocytes.
Ohkura and colleagues, led by Agatha Gluszek, Fiona Cullen and Wenjing Li, found that Sentin-deficient oocytes still managed to form a bipolar meiotic spindle.
And chromosome also more or less okay. It's a little bit different. Chromosome is more compact. So we decided to look at centromeres by in situ hybridization. And then we found these homologous centromeres are not separated in many oocytes.
This suggested that Sentin-deficient oocytes might fail one of the most important steps in meiosis, attaching the kinetochores of homologous chromosomes to opposite spindle poles. Ohkura and colleagues therefore examined whether kinetochores were misoriented in the mutant oocytes.
Then basically we found the kinetochores are asymmetrically distributed as we expected from the fish results. But the more surprising thing is this asymmetrically distributed kinetochores do not undergo the correction, unlike wild type, in which if kinetochores are asymmetrically distributed, then quickly corrected.
To investigate whether kinetochores were misoriented due to alterations in their microtubule attachments, Ohkura and colleagues examined the behavior of GFP-tagged rod. This protein accumulates in foci at unattached kinetochores, but after microtubule attachment, it streams away towards the minus ends of kinetochore microtubules. In wild-type oocytes, rod GFP streams were symmetrical because homologous chromosomes were correctly attached to opposite spindle poles. In Sentin mutant oocytes, however, kinetochores appeared to attach to microtubules even at early stages of meiosis, and the rod GFP streams were largely asymmetrical, indicating that homologous kinetochores often form monopolar attachments to the same spindle pole.
So then we followed from the beginning of nuclear envelope breakdown and spindle formation. And what we found, first of all, in wild-type oocytes, for 1 hour or more, the kinetochore only form a transient interaction with the microtubule. And then in Sentin mutants oocytes, the kinetochore form a stable attachment to the microtubule from the very beginning of the spindle formation.
Sentin therefore seems to prevent stable kinetochore microtubule attachments until oocytes have assembled a proper bipolar spindle. In the absence of Sentin, kinetochores form stable attachments before the spindle is fully formed, increasing the chances of homologous chromosomes attaching to the same spindle pole, resulting in chromosome missegregation and female sterility. Sentin isn't required in somatic cells or the male germline because, unlike oocytes, these cells contain centrosomes and can therefore assemble their spindles faster, reducing the risk of monopolar attachments.
In the beginning of the spindle formation, Sentin localized all over the spindle, probably representing where the microtubule placenta are. And then after spindle bipolarity are established, then Sentin gradually accumulate to the spindle poles. Sentin is a catastrophe promoting factor. So this sending away from chromosome to the spindle poles may stabilize kinetochore microtubule attachment at a later stage of oogenesis. That's just a hypothesis.
In support of this idea, though, a truncated version of Sentin unable to bind to EB1 still localized to spindle poles, but was unable to rescue the phenotype of Sentin mutant oocytes, indicating that Sentin functions at microtubule plus ends to delay the formation of stable kinetochore microtubule attachments before spindle bipolarization. Mouse oocytes affect a similar delay by phosphoregulating kinetochore proteins, though it remains to be seen if they also regulate microtubule dynamics through a Sentin-like protein. But what do Ohkura and colleagues want to investigate next?
So we want to know how Sentin is regulated and how microtubules are finally attached to the kinetochores. As I already told you, one possibility is this Sentin localization to the pole at a later stage may contribute to stable attachment. However, there may be other alternative. Maybe Sentin activity also is downregulated at later stages, or some counteracting activities may be increasing. These are all possible.
In the meantime, however, you can learn more about how the microtubule catastrophe promoter Sentin delays stable kinetochore microtubule attachment in Drosophila oocytes in the paper by Gluszek, Cullen and Lee et al., published in the December 21, 2015 issue of the Journal of Cell Biology.
Ciao.